now's as good a time as any, Mr. Newman. Take her down to periscope depth. Yes. Thanks. Thanks. Well, now's as good a time as any, Mr. Newman. Take her down to periscope depth. Yes, sir. Clear the bridge! Dive! Stand by.
Awesome. Thanks so much. We're live. Yes, we're live tonight. Guys, we've got a really, I think, very important broadcast tonight. We're going to have a lecture with Salvador here. So beforehand, I want to thank him for giving us his time. We've also got John, Jason, and Praise here as well. We're going to be sitting back. If we have any questions, we'll do so. We'll have a short Q&A. After the fact, this lecture is going to be on junk DNA, ENCODE, DNA function. I know Salvador has more of a technical name for it. I'll let him cover that as well. But yeah, we're really excited for this lecture, Salvador. So thank you for giving us your time tonight.
Yeah, thank you. It was awesome. Really appreciate it. I'm really looking forward to it.
Well, if I could share my screen, and I'll just get to it. I won't even introduce myself. I'll just start talking. That's all right with you all?
Yeah,
of course.
It's fine.
At this
Everybody
point on our
knows you.
channel, you're no stranger to our viewers and listeners. So we really appreciate all the work that you're doing and everything that you teach us. So once again, thanks so much.
can you see my screen? It says 4D nucleome, E4 epitranscriptome.
Yes, I can see
Oh,
it.
great. I believe true science is a gift of God. When our Lord and Savior Jesus Christ said, "Consider the lilies of the valley; they neither toil nor spin. Yet I tell you, not even Solomon was arrayed as one of these." I think of the wonders of the cell. Evolutionary biologists like Dan Grauer tell us genomes are mostly junk, especially the human genome. But this presentation will suggest how genomes are amazing machines, thus affirming the sentiments that even the lilies of the field are arrayed in a way far greater than Solomon in all his splendor. So I've opened with a theological thought, but the majority of my formal presentation will be almost pure science in terms of observation and relevant experiments criticizing the claims of Dan Grauer and evolutionary biologists who are on his side. Maybe one way to introduce the theme of this presentation, this is a slight modified quote from the movie Wrath of Khan. And Mr. Spock says to Captain Kirk, he, that is Khan the enemy, he is intelligent but not experienced. His pattern indicates one-dimensional thinking. The original quote was two, but I think you get the idea. So my strategy for trying to show why Dan Grauer is wrong is his thinking is one-dimensional, whereas the modern thinking for the human genome is in 4 dimensions, at least 4 dimensions. If we add other dimensions, it's even more. So we have the 4D nucleome plus the E4 epitranscriptome projects. These are funded projects. These are not just buzzwords. They are funded projects. So this is a quote by Dan Grauer. He said, if ENCODE is right, evolution is wrong. So this kind of sets the stage for what I'll be talking about. I'll actually be trying to argue that ENCODE is right. So independent of evolution, we can kind of discuss whether ENCODE is right, just from the science standpoint. Now, ironically, Dr. Sanford said Dr. Grauer is totally right at his NIH lecture. Slightly different, but that's definitely the sentiment. Just kind of on a superficial level, if we just take the number of gigabases in the human genome, that equates to about 6 gigabits of Shannon information. That's based on the idea that each nucleotide position is roughly 2 bits. So that'd be 800 megabytes. If we take Dan Grauer's view that approximately only 10% of the human genome is functional, that would equate to 80 megabytes. So a rhetorical question, do you think something as complex as a human being can be coded with information as little as 80 megabytes? So that's just something to keep in mind. That's the intuitive thing. That's the first kind of punch I might throw at the idea that the genome's mostly junk. So what is the ENCODE project? It was a follow-on to the Human Genome Project, which completed around the 2000-2001 timeframe. It was headed by Francis Collins. So they found a good swath of, they say pretty much all of the genome was mapped. That is the nucleobases in the human genome. But they didn't know exactly what they did. So they started the ENCODE project, which had a smaller budget, which was actually not, maybe it was just a pioneering project. And it was actually started by Francis Collins. A lot of people resisted him in doing this, but he went forward. So that's also his brainchild. Even though his name's not associated with the ENCODE project today, he was one of the pioneers that motivated its development. So ENCODE has had a follow-on called the 4D Nucleome project. But that's actually that's only part of the picture, because ENCODE has given birth to other projects, such as these. The ENCODE budget, can you all see my mouse here, was a total investment so far of about $500 million. One of the parallel follow-on projects was the Roadmap Epigenomics project, and also known as REMC. That was a $300 million follow-on. I don't know the budgets of these others, but they're throwing serious money at this because they're trying to cure heritable diseases. So this is from the NIH website on the 4D Nucleome project. And these are some things I'm going to cover tonight from the 4D Nucleome. So what did ENCODE actually do? What got all this started? Well, they wanted to know what the genome did, what parts of it did. They just basically had a parts list when they did the human genome project, but they really didn't have a handle on what the individual parts did. So they just started doing these series of experiments, and you have in these blue boxes are the names of the kinds of experiments that they would conduct. And this went on. The people at the NIH would farm this out to laboratories, etc. And they were creating what is known as an encyclopedia of DNA elements. That was their goal. There is another project, not at all related to ENCODE, called the E4. It's called the Enabling Exploration of the Eukaryotic Epitranscriptome, and that is a mouthful, so they just call it E4. E4. So we've heard about epigenetics, which is kind of the extra layer of information that is put on the genome. They're finding there's an extra layer of information also put on the RNA transcriptome. So the genome's DNA, and we have modifications there. The transcriptome is made of RNA, which comes from the genome, but it has modifications, and we call those modifications the epitranscriptome. And here is a little diagram of some of the modifications of the RNA molecules, and there's a whole encyclopedia of these too. And there's even known what is RNA editing. So there are just levels to this. This is probably also just as equally interesting, but we don't have the chemical power yet and the instrumentation to delve into it. But we suspect it's going to be a very interesting world. So this is where a lot of the DNA that we may regard as junk, it will create RNAs that have all sorts of interesting properties. So just bear that in mind. I won't be able to cover it much in this talk. In fact, I doubt I'm going to get through all my slides. And let me check the time. Let's see. Okay, you guys kind of just keep me posted on the time, and I'll just go as far as I can. This was from Dan Grauer's university website. He put this on his university website, and he shows a picture of his granddaughter sticking out her middle finger, and he just says on his university website, that the students and faculty could see, he says, "My granddaughter gives ENCODE the finger." All right, so there's this anger there, and he called. He was calling the people of ENCODE all sorts of names on his Tumblr blog. He calls them crooks. He calls them ignoramuses. He calls one of their heads the scientific equivalent of Saddam Hussein, and probably the ultimate insult, he calls the NIH director, the founder of ENCODE and the Human Genome Project, Francis Collins, he calls him a creationist. So I just had this little video.
It's only a game. Why you have to be mad? So he, Grauer, and some of his evolutionary biology buddies put together this paper criticizing ENCODE. It was just terrible. The tone was vitriolic. It was unprofessional. It was insulting to many. There were probably over 200 researchers involved, from Harvard, Yale, MIT, just all these research institutions, including the NIH, and you're just calling them names. The title of the paper is on the immortality of television sets, function in the human genome, according to the evolution-free gospel of ENCODE. This was very uncollegial on his part, and people had noticed. Another journal called him out on his behavior, and they called him the vigilante, and that was from the journal Science, which is one of the most prestigious scientific journals. And they're noticing this is not right. This is not behavior appropriate to a scientist. Someone can be wrong, but the way to call him out is not the way Grauer is going about it. calling them crooks and ignoramuses. That's just very unbecoming. Just argue the facts. So the way I frame the discussion tonight, this is evolutionary biologists versus medical researchers. This is not about evolutionary biologists versus creationists. This is evolutionary biologists versus medical researchers. This is a fight people like Dan Grauer had started. So what did he have to say? His 2016 archive paper said, if 80% of the genome is functional as trumpeted by ENCODE project consortium, then 45 to 82 deleterious mutations arise per generation. For the human population to maintain its current population size under these conditions, each of us should have on average 3 times 10 to the 19th to 5 times 10 to the 35th children. This is clearly bonkers. Just an amusing anecdote, doesn't have anything to do with this. I was pointing this out on internet discussion forum, and I said, that's kind of bad that a human female has to have 10 to the 35th kids. And someone interjected, what's the problem? What's the problem with that? By the way, the guy who said that is a psychiatrist, and he's an anti-creationist psychiatrist. Sorry, that was an anecdote. Had nothing to do with anything, but just a little humor. So Dan Grauer concludes, if ENCODE is right, evolution is wrong. And as I mentioned earlier, John Sanford said Dan Grauer is totally right. So is the question tonight, is the human 90% junk or not? I gave already one reason that I suspect that it's not. Here's another reason that it started to motivate people. A paper came out. That's actually cited Francis Collins here. Francis Collins' paper in the Proceedings of the National Academy of Sciences, 2009. 90% of disease-associated SNPs, that is single nucleotide polymorphisms, which are mutations, point mutations in the genome, 90% of disease-associated SNPs are located in non-coding regions of the genome. Now, correlation does not equal causation, but intuitively, this is raising alarm bells that maybe we shouldn't call this junk. When I was at the 2015 ENCODE Users Conference, where there are all these scientists learning how to use ENCODE data, and they were talking and they're conferencing with each other, I'd hear one scientist get up and say, yeah, I'm studying this disease or this particular cancer. And what I find is that it's correlated with mutations in the non-coding regions. They could specifically identify non-coding regions. So that was a story over and over and over again. So that's why there's interest in the non-coding regions. So one can imagine the surprise when the ENCODE researchers were starting to publish and say, hey, we suspect strongly, we're confident to declare that we think 80% of the genome is functional. These guys were just doing their job. And then an evolutionary biologist like Dan Grauer, who had no business meddling in medical research, just started calling them all these nasty names. So I said, this is a very interesting development, and it did bother me. It did bother me. I said, this is not good. Excuse me, I'm gonna move. So there is, when we get those outrageous numbers of 10 to the 31st children, it's related to this formula, the Poisson distribution in Kimura's 1966 paper. We're not going to get heavy into the math. In the Q&A, if someone really wants to get into it, we can talk about it. But that formula was indirectly quoted here in Dr. Sanford's NIH lecture. I call it the bonkers formula, the bonkers formula. It is also quoted in other papers, and you see it like in Adam Eyerwalker and Keith Lee's paper. By the way, what I encourage people, creationists, when they're trying to defend Dr. Sanford's work, I encourage them to say, this is not about what Dr. Sanford says. Dr. Sanford is only one among many geneticists. Every top geneticist on the planet that studies the human genome will actually agree that it's deteriorating. That's not isolated. To try to, these internet evolutionists will try to make it out that this is only Dr. Sanford's thesis. It's not. It's many others. It's only fair to point that out. People like Michael Lynch and Alexei Kondrashov. He even wrote a book, The Crumbling Genome. Brian Sykes at Oxford. Gerald Crabtree at the Howard Hughes Institute and also Stanford University. And last but not least, Nobel Prize winner Hermann Mueller. Now, Mueller was the one who came up with what I call Mueller's limit in this journal in 1950. And his limit was the number of mutations. The limit is 1 mutation per individual per generation. There's some nuance between the haploid and the diploid mutation. That's not that important. But compare the limit that he postulated is only 1 mutation per individual per generation. Grauer accepts that he's willing to accept that there are 10 mutations per individual per generation that are functionally compromising. And if ENCODE's right, it's on the order of 80 to 100. There have been suspicions that it could even be in the thousands. But the point is, even the minimum number is 10 times beyond the Mueller limit. Now, I'm not going to mathematically demonstrate the Mueller limit as there's a rich literature that's associated with it. I'm just going to argue from intuition. So just take an intuitive argument for what it's worth. It's inexact, it's simplistic, but it arrives at pretty much the same number that Mueller did. So I hope you guys have had something to eat. These are my some of my favorite foods: tiramisu on the left and a McDonald's bacon double quarter pounder on the right. The ingredients list for tiramisu is eggs, mascarpone, espresso, ladyfinger, sugar, spiced rum, and cocoa powder. Could we, through random mutation and quote-unquote natural selection, evolve it into some other list of ingredients for another recipe? And you all probably know the answer, but I'm just going to walk through this exercise. So, by the way, this is Herbert Spencer on the right. He uses the word, he's the one who introduced the phrase survival of the fittest, which Darwin himself adopted in his later versions of Origin of Species. So let's try to do this by a process of random mutation and natural, quote-unquote, natural selection. So I'm just going to do kind of like a very simplistic population here. So let's say that the original recipe is this, and we'll call it the parent in the first generation, and the parent has children like this, except the children have mutations like that indicated in red. So the problem is, what happens if natural selection takes place? Well, we're going to select only from the best of the worst, so to speak. You know, it doesn't matter what we pick. All of them have some defect. I'm just going to pick child number 2 as the fittest. And child number 2 will have her children. And these children will inherit the parents' mutations, and each of the children will have their mutations. So we could see the mutation is not being filtered out by survival of the fittest, and the mutations will accumulate. There's hopefully no point in continuing. We know the result will be just a total mess. So the question is, could we evolve the ingredients of tiramisu to some other set of ingredients? I hope the answer is clearly no. There's some subtleties to this. If we have an average mutation rate of, say, 1 per individual, that would imply we could have some with no mutations, some with 1, and some with 2. The Poisson distribution covers that. So when we work through the numbers, we end up getting this table here so that we could at least, you know, if we had a certain average mutation, diploid mutation rate, we could estimate how many females at minimum we would need, even assuming 100% efficiency of selection to keep the genome of that species from deteriorating. That was based on Kimura's bonkers formula that I pointed out earlier. So for even with just 1, each female would need to have 5.4 kids, and for 2 it'd be 14.7, so on and so forth. And Dan Grauer pointed out for 80 mutations, each female would need to have 10 to the 31st kids. So I'd like to just pause here if anyone would like to have any comments before I move on. So that's just setting the stage.
And any questions from anybody, I guess, on the panel? Yeah, I'm just fascinated by the talk so far. I mean, just how it sounds like so many people have underestimated the functionality of the genome. I mean, if less than 2% of the DNA actually codes for proteins, you know, what's the rest doing? And kind of has, as you hinted at near the beginning, Sal, like a lot of these non-protein coding RNAs are now known to regulate virtually all levels of gene activity in the genome. I guess one question that I would have is regarding the criticizers of ENCODE, how they will criticize the definition of function that ENCODE used. Because as you were talking about how ENCODE, the results suggest that over 80% of the genome is actively transcribed into RNA, suggesting some type of function and activity. How does one respond to that type of criticism?
What I do, I mean, me personally, I don't get into definitional arguments over function. I really just try to avoid it. I said, this is all a red herring. The point is, do you think it's going to be a good thing or bad thing to have this change? And that's you know, over time. Right. And what I said, I don't even try to argue that the whole genome is functional or 80%. I said, what's so bad about saying we don't know? Is it such a terrible thing to say this is a working hypothesis? Because we have all these medical researchers, like I said, 90% of the heritable diseases are indicated in the non-coding regions. So let's just have a look. Why do they have to be called names? Why do they have to be called crooks and ignoramuses? This is so degrading. And these guys are trying to save our lives. So that's what I'm taking issue with. I said, this is a lot, you know, this whole thing about function, you know, this is maybe not a nice thing to say, but maybe I'll say it anyway. These arguments, when they start, this is like throwing up red herrings when you don't have really a case. Right. When the data are really saying something that should get your attention and they're trying to suppress more investigation. I mean, what is the harm in trying to learn more of how the genome works? And so what if someone is mistaken? It's not like evolutionary biologists have been right all the time. So, I mean, so that's how I would argue it. I say I'm not even going to define function. Let's just keep looking. Let's try to keep understanding. Let's keep running experiments and finding, because we're trying to solve cures here. These are just sideshows. These are just total sideshows that are irrelevant. So sorry, I'm quite a bit more animated today than I was on Friday talking about the origin of life, because I think this is a gross injustice to the scientific enterprise in medical research. Yeah,
I think you put it perfectly. You hit the nail on the head when you say it's a red herring. You know, that's exactly what it is. I mean, it can't be a bad thing that we're finding more function in the genome. And I guess one other question I would have before we move on to someone else who may have a question is, you spoke a lot about, you know, Grauer there and his claims that, I guess with his most recent paper, I think it was in 2017, where he was saying that no more than 25% of the human genome can be functional. Is he saying that based on, like, with the mathematics that he's looking at, is he using like a deep time evolution starting point? Because he more or less is he talking about that in order for human populations levels to be sustainable, no more than 25% of the human genome can be functional because of the effects of harmful mutations? Like, the more functional the genome, the quicker the genome would degenerate, I'm guessing, because the more functional it is, those neutral mutations would be more so nearly neutral. Like, can you expand on that a bit, Salvador?
Unfortunately, I can't, because I stopped reading him. And this is also not a nice thing to say. It looks like the scientific community is not publishing his garbage anymore, really.
That's good.
'Cause he's relegated to publishing on these internet archives, not getting in peer-reviewed journals. And I don't blame editors for not wanting to touch his stuff, because it's not relevant. How does that help us cure cancer or heritable disease or diabetes? Really? I mean, if I went to a medical researcher, is he going to care? And I could tell you, I've hung around enough ENCODE researchers, not ENCODE, but NIH researchers and scientists. They don't care about what Grauer has to say. They never cite him in any of their research or their lectures. He's irrelevant. He's irrelevant. And you know who's actually given him relevance? Unfortunately, it's the creationists. We need to start ignoring him. And unfortunately, I had to give him some space here just to give a little bit more drama.
Yes.
But he's. I'm sorry, I'm going to offend a lot of evolutionary biologists, but their work is irrelevant. It is so irrelevant to these questions. I don't know why these guys are doing all these phylogenies on, this creature evolved from that, when it doesn't really have a lot of relevance to what the mission in medical research or biotechnology. It really doesn't.
So, quick question for you on the phylogenies and such. To me, when I look at that, the entire concept, it's almost like they're coming up with hypotheticals of, oh, well, if all of these changes happened, then it could be this. And is that a fair? I mean, I know there's more to it, but to me, it's at the core. That seems like what is going on there.
I wish I could answer your question. I've given up. I feel I've wasted too many years of my life studying evolutionary biology, and I've studied it also at the graduate level. And I really wish I could have answered your question.
No. The reason I bring that up is as you're going to be, I know you're going to be diving in much more detail on this topic here, and the details here in just a second. And to me, when I listen to, and this is more in the internet atheists debates and such, when I hear them making these arguments, I'm like, you are literally putting forth a hypothetical and then extrapolating based on your own hypothetical situation or scenario. And I'm like, how I can come up with anything that can hypothetically end up in this, especially when, we talk about things of, as you know, I've discussed my backgrounds in technology and software and such. And I'm like, yeah, well, I guess in theory, I could come up with a scenario where all of these different lines of code modified and ended up in a new function. That's irrelevant to whether or not it's a plausible supposition to be making.
And worse, it's not testable. There's no way to really test it empirically. I mean, it may be just the best phylogenetic tree you can come up with, and it sort of, quote unquote, makes a prediction. Like, I could make a prediction. If we evolved from bacteria, there has to be some creature in between a bacteria and a human being. So I'll just pick a eukaryote that has some similar genes. It's like, well, yeah, prediction fulfilled. It's not much of a prediction. I mean, so anyway, I'd like to move on maybe to the more interesting stuff. So thanks for the questions. Could we save it for later if there were any more?
Of course, yeah. And I want to say, I really appreciate you answering those questions, Salvador. So yeah, continue on.
All right, so back to Mr. Spock. He said the opponent is intelligent, but he's thinking in one dimension. So we have to think of DNA in multiple dimensions. And 4D nucleome, as the phrase suggests, is looking at DNA in 3 dimensions and also in the time dimension. And one way to look at DNA, we tend to think of DNA, and it's in the textbooks, is you'll often see DNA is a blueprint for life. Well, really, it's a blueprint for the proteins of life. And I actually people are beginning to suspect that a lot of the heritable information is in the cytoplasm and even in the membranes, but that's just a story for another day. I mean, it really could be that DNA is just scratching the surface of the information capacity inside the cell. Because if you include the cytoplasm, it's going to be gigantic, especially the sugar molecules. And they're talking about sugar code. People can Google the sugar code. So we may have only hit the tip of the iceberg. But that being said, for the nucleome, if we look at DNA as a scaffold, as a material, as a physical material to park molecular machines, we begin to see how things could be functional. Because up until then, we've just been thinking of DNA as coding for proteins, maybe some regulation. But now when we start to think of it as a scaffold, it's different. Another metaphor, and as imperfect as metaphors are, these are the best I could come up with. We can also think of DNA as designated parking spaces with addresses. So you see these parking spaces have names to them. So the spelling of the DNA letters is important because that serves as an address. So think of it as addressed spaces. In DNA world, we call that names, we call these things motifs. So these are motifs that machines will search out for to park the machine. And I'm going to illustrate that. Most of the panel here probably is familiar with chromatin, but for the listeners here, I'm going to have to cover some basics about DNA. In organisms like humans, eukaryotes, we have an architecture like this called chromatin. So this is how our DNA is organized in eukaryotes. Bacteria do not have this form of organization. So here we have a chromosome. And if we start to pull it apart, it unwinds into these wires or these strands called chromatin. And chromatin is composed of DNA. Excuse me. It's composed of DNA that wraps around this bead-like structure called histones. So DNA wraps around things like histones. So it's like a bead. People will refer to it as beads on a string, where the string is DNA and the beads are the histones. And you can actually see here the DNA expanded out. Everyone okay with that? Now feel free to stop me as I go through these slides because they are technical. If I miss something, please ask me a question to clarify, because I sometimes will mistake things. So we have this, you'll notice this, it says here, this methyl group. That is a modification on the DNA. It's like a little post-it that's stuck on the DNA. That is what we call, that is one form of the epigenetic mark. It's an epigenetic modification. It's just like adding a little extra chemical there to the DNA. That's one form. Another form of the epigenetic mark is changes. We can add little chemicals, like little post-its, to the tails of these histones. So you have these histones here, and you have these little tails protruding, and we can add chemical markings to these. These are epigenetic markings. Let me cover the methylation marking here a little more. And this is all related to the discussion about making if DNA is functional. So we have here a cytosine molecule, and it can be modified by a methyl group. There's some question whether some of the other molecules can be modified too, but so far the cytosine is like the dominant one in our literature. So we can add a methyl group. And so this is, if you imagine the red here, that's the DNA double helix, and you can have these little methylation marks. So this is reminiscent of random access memory. By the way, the name epigenetics, I believe it came from this gentleman here, Waddington. And here's another picture of some epigenetic changes. I will say this, I don't like the word epigenetics anymore. At the time that it was coined, they didn't have all the chemical details. So now that we have the chemical details, we don't have to call it epigenetics anymore. We can say, okay, this is a methyl modification on the cytosine, methyl modification on DNA. Then there's no confusion about what you're saying. But this is a little introduction to what epigenetics is all about. Unfortunately, there are a bazillion definitions of epigenetics, and they don't agree with each other. So I'll try to just be more specific. So I'd like to show what happens with these methylation changes. This is a box A here. They're able to make these very interesting graphs where they, I believe they use luminescent proteins that would, or antibodies that would bind to the methylation marks, and then you can illuminate them. So we have a zygote here, and it's at the stage where the the dad's DNA, the paternal DNA, hasn't yet mixed with the maternal DNA. And you can see the methylation marks by the illumination there. And that's at 3 hours from after fertilization. At 6 hours, it looks like this. But come at 8 hours, notice the paternal DNA has erased. It's like done a reboot of its methylation marks. See that? Hope that's clear. And then sometime later, it continues. And then when the cell enters the zygote enters the metaphase, you could start to see I guess these are the chromosomes starting to break apart, and they're going to do their thing. Ignore the middle panel. That's just the control region. That kind of makes the diagram confusing, unfortunately. But then when we enter the 2-cell phase, you could see the changes. You could start to see the maternal DNA that is methylated, merging with the paternal DNA. And this is the 2-cell phase. We get to the 4-cell phase, and you could see the DNA, the methylation marks turning off. So what does that remind you of? That reminds me of random access memory. It's almost like a punch card. In fact, some of the diagrams that list the methylation marks between cells looks like this. I got this from, I know some people will say, well, where's your citation? Just Google it. If you search far enough, you're gonna keep getting all sorts of material. And I often quip, whatever I say right now is probably 5 minutes obsolete because the field is moving so fast. So you could see that the methylation markings are different between different kinds of cells. And from the Stem Cells Handbook in 2013, they use the word random access memory for epigenetic memory, random access memory. So I used to get flagged on that. They used to say, Sal, you're just making stuff up. And so I Googled real hard and had to look, and there it was. Other biologists were thinking the same thing. So great minds think alike. I kind of rubbed it in their face. So here is also some visualization of how this epigenetic memory, this random access memory. So this is really amazing. DNA is considered like a read-only memory, but there's a layer of random access memory on top of it. So we have 3-year-old twins here, and these are their methylation marks. And then we have 50-year-old twins, and you could see the change in their methylation marks. So I'd like to pause if anyone had any questions or comments. I hope those were interesting pictures. I don't know if you've seen them before.
No, I think it's, once again, it's so much fascinating. I find the function of the genome so fascinating, just as you were talking about chromatin, how these non-protein coding RNAs from introns actually influence gene expression by modifying chromatin, for the audience sake, as you explain that complex combination of DNA, RNAs, and proteins, right, that make up chromosomes. But for me, no specific questions, just once again, so much fascinating information. And John, Jason, Praise, any questions or comments from you guys?
Yeah, I'm just enjoying listening. I had a question, but I'd forgotten that as usual. But yeah, I know I'm really enjoying it. I'm really enjoying watching and learning about this. And there's a lot of new terms in there that you know, that I want to write down later and get timestamps and stuff. So I was really enjoying it, Dr. Cordova. You're doing a really good job.
Well, thank you. I'm only Mr. Cordova right now. But I do have 5 degrees though, so I have kind of a broad education, not specialized.
Oh, Dr. Cordova, sorry.
Mr.
Yeah.
Cordova is fine. I'm not offended.
No, I'm sorry.
No need to apologize.
If there's no more questions, we can let. I do have a question. Do you prefer to be called Salvador or Sal?
Just call me Sal.
Sal. Okay.
Okay.
It's nice to be called Sal. My detractors call me all sorts of names, so it's refreshing to be called.
Same here.
Well, you're doing a great job so far, Sal. You can continue whenever you're ready.
So this is chromatin. So I covered the methylation. That's one form of epigenetic change. The next form of epigenetic change will involve the histones. So these are the histones. These are beads on the string. I like this beads on a string. So just think about it. This is random access memory with beads on a string. I'd like to compare some of the historic man-made memory with this. So on the left side is what I call God-made random access memory. This is a picture of chromatin right here in an electron microscopy. So you see the beads here, like that. And you can barely see the wire. That's the DNA that's between them. So that's beads on a string. That's God-made memory. I'd like to contrast that with man-made memory. There was an old 1950s core memory that used magnetic core. It still sort of looks like beads on a string. And even farther back in ancient times, we had kind of beads on a string. There's sometimes they actually did use strings, not just wires or sticks, beads to have memory. That's an abacus. So that's kind of, I'm a very visual person, and so I just found this compelling, personally. So I call that God-made random access memory. This is another depiction of chromatin. I'm sorry?
I said, yeah, wow. I said, that's amazing. You know, that the way that's set up like that just couldn't be anything other than made by a creator.
Isn't that cool? That just
yeah, it is. It's amazing.
Yeah, the
yeah,
Lord
it's
yeah. I think the Lord has a sense of humor. I just wow.
Yeah. Yeah, sorry.
Well, we'll go on. I'm glad you're enjoying it. So the reason I had this other picture, I wanted to highlight some of the more fine features of the histone. They're actually, the histones come in pairs. They're identical. And so there are 4 identical pairs to make 8 histones, but we usually kind of tend to think of them as only 4. But they're actually 8. And I'm just gonna take what's in the circle, and I'm gonna kind of expand upon it. So these are the histones, but I'm focusing on the tails. I don't know if, let me go back. So you see these tails out here? This diagram is going to expand the tails a little more. And so like you have histone 3 here, and you can see all the different kinds of positions here on the tail, and they can have these little post-its, chemical modifications. The Me is for methylation, P is for phosphorylation, AC is for acetylation. I don't see any, there are not too many ubiquitinations, but they do, oh, here's one UB modification there. And just for completeness, I want you to see how the histone is spelled here. This histone 3, let me go back one. So you have this histone 3. I'm going to spell the protein out in terms of its amino acid letters. It's only less than about 140 letters. And the numbering of the histones is a little unfortunate. It's a little different. Everyone has their own coordinate system. But I'm going to number, this histone is, this K here, which corresponds to a lysine amino acid, is position 27. So I just go from 0 all the way up to 27. That corresponds to this one here. Do you see that? I have the orange mark here and the orange mark there. I'm going to show all the machinery that it takes to add a post-it note to modify that. So it's just like flicking a bit. So this is also another form of random access memory. We can be flipping bits, which is really cool. Does that mean chromatin is a computer? Some people have suspected that it is one of many computers in the cell. And indeed, they borrow language from computer science. So when we're flicking all of these, we're making these chemical modifications here, we say we are writing, we are erasing, or reading the mark. And they have all these chemicals that do it. So we have readers, writers, and erasers, very similar to the computer read-write heads in our machines, like say like a disk drive, except that the difference is in biology, you have multiple read-write heads that are just floating around inside the cell. And what's really amazing, it has to navigate to the right position. And I'm going to show that just for completeness for the chemistry nerds out there. This is how the methylations work. I just threw that in there, but we don't need to go there. We can kind of abstract out all the details. So again, we have we have a chromosome, and we have these, we call this complex, we have the histones with the DNA wrapped around. By the way, we also call this little unit a nucleosome, nucleosome. And by the way, that sounds very similar to nucleome. So when you hear 4D nucleome, these are similar words. So how does it navigate? And these readers, writers, and erasers have to navigate to the right nucleosome particle, the right histone, the right section of DNA. So I'm going to show that. So let's start with chromosome 12 here. Chromosome 12, let me highlight it. Chromosome 12 has a section of DNA that we call hot air. So just imagine we found this, we are looking at chromosome 12, and we found DNA that looks like this. And we have a name for it. It's called the hot air link, long non-coding or long intergetic non-coding RNA. It's actually a DNA that codes for an RNA, so we call it hot air RNA. So how does this work? So remember, I'm just going to flip one bit. I'm going to flip one bit on a histone. So we start with the hot air link RNA. It comes out of the chromosome 12. It somehow navigates to chromosome 2 through all that colloquially what I call Brownian motion. That's not the correct formal term, but it is a it's close enough. It illustrates the point. So it's going to somehow navigate without any active propulsion in the nucleus, in the cell. It's going to find its way to chromosome 2, and we actually don't know how it does that. That's so far it's like black magic. We have some idea, but right now it's pretty mysterious how it can do that. That's an amazing problem in biophysics. So that hot air RNA, it'll find a section on chromosome 2 that has a place just for it. It's like a named address. I'll just call it the hot air space. So that's what I'm trying to illustrate here. There's a parking space just for it called hot air that corresponds for the hot air RNA. When it arrives at chromosome 2, it'll trigger the assembly of this massive complex here called the polycomb repression complex 2. Sorry, I'm losing my voice. I'm going to have to take a little break here and let you guys talk. The black wire here is the DNA. That's DNA. And then these are the histones here. So it's going to find the right chromosome. It's going to find the right parking lot on the DNA. It's going to find the right section of DNA. And it's going to find the right histone. It's going to be histone 3. And it's going to find that methylation mark of position 27. It's going to have all that machinery. Isn't that cool?
Yeah. Yeah, it is. Yeah, it's
So
amazing. I mean, like, sorry, Stanley, go on.
Go on. And you were Jared? Did I get your name
Jason.
right? Jason, I'm sorry.
Yeah.
Jason.
Yeah, it's amazing how all this stuff, links together like this and works in conjunction with each other. It's doing stuff in the genome. The genome's working. I mean, the genome works on first, second, and third dimensions change over time to make the genome 4D, and just the unbelievable complexity and just in a so-called simple protein is mind-blowing. It's and bits of proteins and just even piece. I mean, for these things to be like this, is for anybody to say that just happened like that, like a friend of ours, Neff, always says, Nephilim Free. He always says they'll look at a pile of compost and they'll say a pile of compost is designed, but then they look at something like this and they say it's an accident.
So as you're preparing to move on here, you've mentioned the term machines throughout your entire discussion. Something I've run into on a very regular basis when I make the argument about the nanotechnologies and nanomachines that are enabling this read, write, edit process you're talking about. It's immediately dismissed as nothing but just random chemical reactions. Is that no, we're talking about real life nanomachines. Is that premise accepted inside of academia, that yes, these are molecular machines that are doing direct functions rather than just being this, random chemical reaction that's somehow resulting in these things like methylation?
If you're at the University of Houston and Dan Grauer's department, yes, it'll be a random reaction and of no consequence. But what's happening is we're actually beginning to correlate it to changes. So that little change that I was describing, that correlates to changing the quality of the skin. So depending on how this is flipped, you'll get soft skin like in an eyelid versus hard skin like you get in the soles of your feet. It changes. So these changes matter. And just because we don't know what it actually does it right now, because we're seeing all these enormous number of computations being done, we don't know what it's actually doing in terms of how it develops the organism. But it takes a lot of research, it takes hundreds of people to get a really good definitive answer just for that one. I'll tell you, this modification here is famous. It took at least a year and maybe a staff of 20 people to figure it out. And it was really hard work. And that's just one modification on one histone in the Hox cluster. We don't know, there's so many other regions of DNA where you have other histones, and we don't know what those do. So there's suspicion. So sometimes they're dismissed, but you know how they stop being dismissed? By research. When an experiment proves that this is what it does, the people are silenced. I wanted to say, and I'm just going to wax a little philosophical here because this relates to the book I'm writing. The way that we actually started to learn about this was studying fruit flies. Fruit flies ended up giving us an understanding for how the human body worked. So how the fruit fly changed its, it had a number of like bristles or something. The way we could change the number of bristles was changing the corresponding histone here, but it gave us a clue of how all of this machinery worked. So that's why we call it the polycomb repression complex. It affected the flies, the bristles on the fly's legs or something. And yet it has a correspondence in humans, but it's like, again, this is theological, philosophical. It's like God gave us all these little model, we call them actually model organisms. That's why it's like, why are these medical researchers trying to solve human cancer by studying fruit flies? It's like, well, we have all these organisms that are simpler that we can kind of break apart and blow apart and do all these cruel experiments that we would never think of doing to humans. They're just like gifts. So all these patterns of similarity are gifts for medical research, so we could understand we're fearfully and wonderfully made. So that's my little mini sermon. So this is how some of these discoveries were made. So that's how it may be dismissed today. And I will say, even 12 years ago, when people were saying this was all junk, I had no answer on the internet. But now it's so easy to fight them because research has advanced in 12 years. So that's how I would deal with that argument when they just dismiss it. I'll say, well, I'm going to wait on the Lord. There'll be something coming up because it's to His glory to reveal this eventually. So this has been hidden for 2,000 years, or ever since time began. We didn't know all these miraculous things. Now in the present day, we're learning it. God stored it up for us in the 21st century.
Well, I find it so fascinating that I guess the DNA in general, but especially the non-coding regions of our DNA, as John was talking about, they do function much like a computer system. And these, I guess the deniers, I find that the ones that say, okay, the majority of our genome must be junk, no more of it can be functional than 25%, as you've stated, Sal, it hinders scientific advancement and it hinders medical advancement. And typically I'll ask those types of people that if all of this is truly junk, if over 75% of the genome is junk. Why is the cell even bothering with it? Because obviously that type of activity would be wasteful of energy and resources, and natural selection should have eliminated all of this wasteful product so long ago. You know, do you find that there could even be an answer to such a question like that, Sal?
No, I don't. I try not to answer them anymore, because they don't even have the same story, because some people say this is neutral evolution, others will say selection. So they can't even get their story straight. What's the point, you know? And whereas what ended up happening to me, you know, it was really part of my job to study this. I said, what have I been missing out on? When I saw this like 4 years ago, I wanted to say hallelujah in class, but I couldn't. And then I tell my friends, I said, wow, this is amazing. We never see this discussed on the internet, if you know. And so now this is why I'm very grateful for you guys. You've given me an opportunity to share this, because sadly,
we're enjoying it.
Yeah,
we really appreciate it.
We're really enjoying it.
It's amazing.
Sorry,
no problem. I find it amazing because what we are dealing with is a highly compressed DNA code that's beyond the best computer code. But I guess those that reject that type of thought process, they are hindering themselves from such fascinating discoveries. Because just like you said, this has been hidden for 2,000 years, and just maybe 20 years ago, 30 years ago, a lot of the pro-evolution arguments had to do with the junk areas of our genome, pseudogenes, ERVs, the ALUs, for example. But now all this research, paper after paper after paper, suggesting that these aren't just leftover remnants of viral infections or genetic mistakes. I mean, these are functional DNA elements, oftentimes important to sustain healthy life processes in the cell. So we can bow down and say, you know, praise God for this. But the ones that want to reject that idea, yeah, they're just hindering themselves and hindering scientific advancement, mostly.
you know, my favorite.
No surgeon ever went to a book of evolutionary origins to brush up on surgical techniques before a difficult operation. You know what I mean? Like, it's so it's ridiculous. It doesn't help anything. It's just a huge waste of money. I mean, billions and billions of dollars that could be put into researching actual cures and doing actual good in the world, instead of wasting it on, you know, rubbish, you know, fairy tales.
Yes.
And yeah.
Exactly. So one of my favorite proverbs is, it's the glory of God to conceal a matter. It's the glory of kings to search out a matter. God has concealed this for us to discover. It's just amazing. So we live in a very, very special time. I mean, Christians would have longed to have seen what we see. We are so blessed. If I can, I'm going to point out some things.
Of course, take your time, Sal.
So the importance of the DNA sequence is that it gives a name. It gives names. So the sequence is important. And this will relate eventually to repetitive elements, but I'm going to point that out. And it's very important for addressing that we have the right spelling. So the spelling matters. And you can kind of guess that how do the readers, writers, and erasers, how do they find the right location on the protein? It has to be spelled right, because that's how it's going to navigate there, because otherwise it's going to grab another protein. So there has to be some uniqueness in the addressing, and that's why there's certain sections of protein that really, really, really count. You cannot be modifying these willy-nilly, because if you start to change even the amino acids in between, like between number 28 and 38, that could mess up the ability of the reading, writing, and erasing machines to locate. See, I'm just pointing, that's looking farther down the line. So the sequence is important. Yeah, here are the readers, writers, and erasers. Now, I showed a really simple chromatin computation with the, I was just modifying this one, okay? I've seen the diagrams get ever more complex. So I saw one like this. I said, evolutionists wanted to debate me on the emergence of eukaryotes. So I threw this diagram up. I said, okay, So archaea has kind of like a pseudo chromatin, but eukaryotes, it's really special. For eukaryotes to do DNA transcription and translation, they have to do stuff like this. That's not even doing, that's just to getting it off the ground, because if there's a double-stranded break in DNA, it has to go through all these histone modifications and coordinate them. Huge. And I said, do you really want to debate me on this? You could have a system that was just all DNA, and then now you have chromatin. You have to have all these machines to do all the what they call remodeling and all the cross signaling. That debate lasts about 1 minute. Okay, so that's why.
All right, guys. I'd like to thank everyone for coming out. Some of the rendering of this recorded video was not as good as I was hoping, and so that's motivating me to redo some of this. So what I'm going to do, since SFT is hosting his Jason Lyle show, I'm going to go ahead and shut down, and please head over to his stream. Take care and God bless you. Thank you for joining us this day.